Gene therapy aimed at upregulating HDRT, the gene encoding the catalytic subunit of telomerase reverse transcriptase, represents one of the most ambitious and cutting-edge approaches to telomere restoration and potential lifespan extension. At the heart of this strategy is the idea of reawakening the body's dormant capacity to rebuild telomeres in somatic cells, which typically experience progressive telomere shortening with each cell division. In early animal studies, particularly in mice, upregulating HDRT has been shown to not only lengthen telomeres but also improve tissue function, delay age-related degeneration, and, in some models, significantly extend both health span and lifespan. These outcomes suggest that HDRT-based therapies may hold extraordinary potential for slowing the biological clock at the chromosomal level. In most adult human cells, telomerase is either completely inactive or tightly regulated, in part as a natural safeguard against uncontrolled cell growth. The enzyme is fully active only in select cell types, most notably stem cells, germ cells, and certain immune cells, where it is crucial for long-term replication and regeneration. Reactivating HDRT in somatic cells using gene therapy essentially aims to restore the ability of these cells to preserve telomere length indefinitely, potentially overcoming one of the primary limits to cellular longevity. The theoretical benefits are profound: improved regenerative capacity, protection against senescence-related decline, and the ability to maintain tissue integrity over a longer period of time. Mouse studies using viral vectors to deliver HDRT into specific tissues or systemically have yielded impressive results. In older animals, transient expression of telomerase through controlled gene therapy has been shown to rejuvenate aging organs, reverse certain markers of cellular aging, and extend lifespan without increasing cancer incidence, provided the therapy is carefully timed and targeted. These outcomes suggest that under the right conditions, HDRT upregulation can be safely harnessed to counteract the effects of telomere shortening without destabilizing genomic integrity. The promise of such therapies for humans is immense, particularly in areas like regenerative medicine, treatment of age-related degenerative diseases, and immune restoration. Potential applications could include reversing bone marrow exhaustion, enhancing skin and vascular repair, or restoring function to aging cardiac or neurological tissues. In theory, a single intervention to selectively enhance telomerase activity in key cell populations could translate into years of improved health, greater resilience to disease, and a significantly delayed onset of age-related decline. However, despite these exciting possibilities, HDRT gene therapy still faces significant challenges, chief among them, the risk of oncogenesis. Because telomerase activity is a hallmark of over 85% of human cancers, any therapy that increases its expression must be extraordinarily precise and tightly regulated. The danger is that upregulating telomerase in a precancerous or genomically unstable cell could enable that cell to bypass the natural limits of replication and become malignant. This risk has led to understandable caution in translating animal successes to human trials. To address these concerns, current research is exploring more refined delivery systems and conditional gene expression models. For example, using inducible promoters, tissue-specific targeting vectors, or self-limiting expression systems allows for greater control over where, when, and how long telomerase is activated. Some studies are even looking at episomal approaches, non-integrating vectors that do not alter the host genome permanently but can still transiently express HDRT to achieve telomere restoration without lasting genomic changes. These techniques, while still experimental, represent an encouraging direction towards safe clinical application. Ethical considerations also come into play, particularly around potential uses in healthy individuals or for non-therapeutic enhancement. The line between treatment and enhancement becomes blurry when considering telomerase gene therapy for the purpose of slowing natural aging. Questions around accessibility, equity, and long-term risk must be considered carefully as these technologies progress. Still, for individuals suffering from premature aging syndromes, severe stem cell exhaustion, or tissue-degenerative diseases, the ethical justification for HDRT-based therapies is strong and increasingly supported by data. In summary, gene therapy for HDRT upregulation represents a bold frontier in the science of aging. It leverages one of the most fundamental determinants of cellular lifespan, telomere length, and seeks to intervene at its genetic root. While significant hurdles remain, particularly around safety and long-term regulation, the proof of principle has already been demonstrated in preclinical models. With continued advancements in vector design, gene regulation, and targeted delivery, telomerase gene therapy may eventually shift from a futuristic concept to a cornerstone of regenerative and anti-aging medicine. It reflects the profound power of modern biotechnology, not just to treat disease, but to reshape the very boundaries of human aging and vitality.